Since the times of Darwin, zoologists have reported that males of a species are more morphologically variable than are females. That is, a randomly selected sample of males are believed to exhibit more variability amongst themselves in terms of, for example, their weight, than are a sample of females. This phenomenon is termed greater male variance, but the extent to which it applies across traits and different species is far from clear. Indeed, some traits show the reverse pattern of greater female variance. Arguably, the most straightforward hypothesis to explain greater male variance and greater female variance is that the larger sex of a species will be more varied because of a simple one-to-one mathematical coupling between the size of the measures taken and their spread. This would predict a unitary relationship across species between mean body mass and the standard deviation of body mass, where proportional changes in the standard deviation follow from proportional changes in the mean. It is possible then that Darwin and others commonly observed greater male variance because in most of the species they happened to study, males are physically larger. So, when greater variability in males is observed, is this associated with males being bigger, and is greater female variability observed when the female is the larger sex? We ran analyses to investigate this question based on the dataset used by Tombach et al. With a few tweaks and additions. We ran a phylogenetic generalized least squares analysis on 336 species representing 6 orders of mammals. Across orders, the relationship between standard deviation and mean body mass did not statistically differ from unity within either sex in any of the orders. Arguably then, the parsimonious interpretation is that the scaling of variation to size is close to unity for both sexes. Our data suggests then that rather than body size variability being related to sex per se, for example that males are more varied than females, body size variability is more consistently related to sex differences in body size. In other words, the larger sex of a species is the more varied one. However, in an analysis of the degree of greater male variation or greater female variation against the degree of sexual size dimorphism, the overall relation was statistically greater than 1. And at the order level, 2 orders, Chiroptera and Rodentia, bats and rodents, had slope values that were statistically greater than 1. These hyperallometric relationships suggest that greater male variation and greater female variation are sometimes driven by other factors alongside sexual size dimorphism. So, we conclude that often the single driver of greater male variance and greater female variance is simply differences in trait size between the sexes. Nonetheless, our analyses hint that in some mammalian orders, other biological processes also influence differing variation between the sexes. Discussion of what these processes could be, along with further analyses of the data, can be found in our paper published at Biology Letters. You can download the paper at the journal's website, or ask for a copy by contacting the corresponding author, Halsey.